Hi, my name is Maryann and today I'm going to talk to you about early embryonic development and the hormonal control of implantation. So today we're going to be discussing early embryonic development and the hormonal control of implantation of the embryo. All the information I'm going to share with you today is from Silverthorne, 8th edition, chapter 26. Let's start this lecture by looking at the anatomy of the female reproductive organs. We are going to focus on the internal reproductive organs of the female since that is where embryonic development takes place. The four tissues or organs I want to talk about are the cervix, fallopian tubes, uterus, and ovary. I have them circled on this diagram for clarification. Now let's talk about the cervix. The cervix is the neck of the uterus which opens into the upper end of the vagina. The cervical canal is lined with mucus glands whose secretions create a protective barrier between the vagina and the uterus. There's a narrow opening of the cervix that allows sperm to pass through in order to fertilize a potential egg. This potential egg would be lingering in a fallopian tube. Fallopian tubes are about 20 to 25 centimeters long and about the diameter of a drinking straw. Their walls have two layers of smooth muscle, longitudinal and circular, similar to the walls of the intestines. A ciliated epithelium lines the inside of the tubes. An egg is transported down the fallopian tubes towards the uterus by a fluid movement that is created by the cilia and aided by muscular contractions. Fertilization can occur if sperm moves up the fallopian tube while an egg moves down towards the uterus. The uterus is a hollow muscular organ that's slightly smaller than a woman's clenched fist. This is where fertilized eggs implant and develop during pregnancy. It is comprised of three tissue layers: an outer connective tissue covering, a middle layer of smooth muscle called the myometrium, and an inner layer called the endometrium. Now let's talk a little bit about the ovary. The ovary is an elliptical structure that's about 2 to 4 centimeters long that produces eggs and hormones, and has an outer connective tissue layer and an inner connective tissue framework known as the stroma. The outer layer is thick and called the cortex, which is filled with ovarian follicles in various stages of development. The smaller central portion of the ovary is called the medulla, and that contains nerves and blood vessels. Now let's take a closer look at the uterus and ovary. Starting with the uterus, we see that it has 3 layers of tissue. Like I mentioned in the first slide, there's an outer connective tissue covering, a thick middle layer of smooth muscle known as the myometrium, and an inner layer known as the endometrium. The endometrium is made up of an epithelium with glands that dip into a connective tissue layer below. The thickness of the endometrium varies depending on what part of the menstrual cycle a female is at. This is where the cells of the epithelial lining alternately proliferate and slough off, accompanied by some amount of bleeding, which is known as menstruation. The uterus is also the place where implantation takes place, which we will talk about later in this lecture. The ovary has 2 main parts, like many organs in the body: the cortex and the medulla. The medulla is also called the stroma here, as it contains nerves and blood vessels. In the cortex of the ovary, which is the outer part, we can see that this is where follicular development occurs. The diagram on the slide helps to show us the different stages of follicular development. It goes through a cycle to release an egg, or what is scientifically known as an oocyte. First, it goes through the follicular phase, which is a period of follicular growth in the ovary. This part is the most variable in length, as it can go on anywhere between 10 and 21 days. Then we have the ovulation, and this is when one or more follicles have ripened and the ovary releases either one or multiple oocytes. And finally, we have the luteal phase. In this phase, we have a transformation of a ruptured follicle into a corpus luteum. The corpus luteum secretes hormones that continue preparations for pregnancy. Now that we know the basics, let's dig deeper into the development portion. Embryonic development starts with fertilization of an egg or oocyte. The first sperm that fuses with the egg is the one that fertilizes it. For fertilization to occur, the sperm must go through its final step of maturation, which is called capacitation, which helps the sperm to swim rapidly and fertilize an egg. This process changes lipids and proteins of the sperm head membrane. Capacitation occurs in the female reproductive tract once the sperm goes through the cervix. An egg can only be fertilized for about 12 to 24 hours after ovulation. The sperm binds to the epithelium of the fallopian tube while waiting for a chemical signal from an ovulated egg, and the sperm can remain viable in the fallopian tubes for about 5 to 6 days. Fertilization occurs in the distal part of the fallopian tube. In order to fertilize the egg, the sperm must penetrate both an outer layer of loosely connected granulosa cells and a protective glycoprotein coat called the zona pellucida. To get through these layers, the capacitated sperm has to release powerful enzymes from the acrosome in the sperm head, and this process is called acrosomal reaction. The enzymes dissolve cell junctions and the zona pellucida, which allows the sperm to enter the egg. The fusion of the sperm and the egg triggers a chemical reaction called the cortical reaction that disregards the other sperm. In this reaction, membrane-bound cortical granules in the peripheral cytoplasm of the egg release their contents into the space just outside the egg membrane. These chemicals rapidly change the membrane and surrounding zona pellucida to prevent more than one sperm to fertilize the egg. To finish fertilization, the fused section of sperm and egg membrane opens and the sperm nucleus sinks into the egg cytoplasm. This signals the egg to resume meiosis and complete its second division. This final mitotic division creates a second polar body, which is ejected, and this is when the zygote is a full set of genetic material. It has 23 chromosomes from the sperm and 23 chromosomes from the egg. When an egg becomes a zygote, it begins mitosis and slowly makes its way from the fallopian tube down to the uterus where it'll implant. Now that we know how fertilization happens, let's take a look at how embryonic development occurs. It takes about 4 to 5 days for the dividing zygote to move down the fallopian tube and into the uterine cavity. The hormone progesterone helps the smooth muscle of the tube relax and transport the zygote along slowly. When the zygote reaches the uterus, it's a hollow ball of about 100 cells called a blastocyst. The outer layer of the blastocyst will become the chiron, which is an extra embryonic membrane that will enclose the embryo and later form the placenta. The inner cell mass of the blastocyst will become the embryo and the other 3 extra embryonic membranes. The other 3 extra embryonic membranes are the amnion, which secretes amniotic fluid where the developing membrane floats, the allotonus, which becomes part of the umbilical cord that links the embryo to the mother, and the yolk sac, which degenerates early in development. The blastocyst implants into the uterine wall about 7 days after fertilization. The blastocyst secretes enzymes that allow it to invade the endometrium, and the endometrial cells will then grow around the blastocyst until it's completely engulfed. The blastocyst will continue to divide and eventually become an embryo. The cells that will become the placenta form a finger-like chorionic villi that penetrate into the vascularized endometrium. Enzymes from the villi break down the walls of maternal blood vessels until the villi are surrounded by pools of maternal blood. The blood of the mother and embryo don't mix, they just exchange things like nutrients, gases, and wastes across the membranes of the villi. Many substances move by simple diffusion, but some of them, like maternal antibodies, have to be transported across the membrane. Now let's talk about how hormones play a role in implantation of the embryo. When the blastocyst implants in the uterine wall, the placenta begins to form and the corpus luteum nears the end of its 12-day lifespan, unless the developing embryo sends a hormone signal to the corpus luteum. The placenta also secretes a lot of hormones that prevent menstruation during pregnancy. Some of these hormones being human chorionic gonadotropin, human chorionic somatomammotropin, estrogen, and progesterone. In early pregnancy, the corpus luteum remains active because of hormone chorionic gonadotropin, or HCG, which is a peptide hormone secreted by the chorionic villi and developing placenta. Because of HCG, the corpus luteum keeps producing progesterone to keep the endometrium intact. By the 7th week of development, the placenta takes over progesterone production, and the corpus luteum is no longer needed, and this is when it finally degenerates. The production of human chorionic gonadotropin by the placenta peaks at 3 months of development, and then it diminishes. Another function of HCG is the stimulation of testosterone production by the developing testes in a male fetus. HCG is also what is first detected by pregnancy tests. Now let's talk about human chorionic somatomamotropin, or HCS. This is another peptide hormone produced by the placenta, which is also called human placental lactogen, or HPL. This alters the mother's glucose and fatty acid metabolism to support fetal growth. Maternal glucose moves across the membranes of the placenta by facilitated diffusion and enters the fetal circulation. Another function of this hormone might be to help mothers' breast development and milk production. However, some women who have a genetic defect and don't make much of this hormone are still able to develop breasts and have milk production. Estrogen and progesterone are the two hormones that are produced continuously during pregnancy, first by the corpus luteum under the influence of HCG, and then by the placenta. With these two steroid hormones being circulated in high levels, there is feedback suppression of the pituitary throughout the pregnancy, which prevents another set of follicles from beginning development. Estrogen is important for the development of milk-secreting ducts of the breasts, and progesterone is essential for maintaining the endometrium, and it also suppresses uterine contractions. Without all these hormones, a viable pregnancy wouldn't be sustainable. Thank you for listening to my lecture about embryonic development and hormonal control of implantation.